Awesome. All right. And that's terrible. That's right in the middle. Okay. So here we are on our 25th chapter. Well, it's not really true, but we got what, 3 more chapters after this one. But the next 3 chapters are all about cellular metabolism, particularly DNA metabolism, RNA metabolism, and protein metabolism. So we're taking our central dogma from the previous chapter and looking at it. So you would think, that this chapter would not be terribly long because it's only got 3 learning goals, right? DNA replication, DNA repair, and DNA recombination. 3 simple topics, except they're each about like 50 to 60 slides. So that's just, yeah, they're ridiculously long. All right, but let's talk about what we mean by DNA metabolism here. So as opposed to our energetics of the cell and producing ATP and our citric acid cycle, all that good stuff here, now we're going to be talking about DNA metabolism in terms of stable storage of genetic information and then making that available to future generations of the cell. All right, so basically before cell division, we got to make a copy of the DNA. Oh, I like my finger gestures in here. Yeah, all right. Finger guns. No, finger DNA. But we want to make a copy that's pretty much as close to the original as we possibly can. And the thing I always like to joke about is how good is DNA replication? Well, imagine you copied your textbook, that whole Lehninger 1,200-page book or whatever by hand without making any mistakes. That would be, an order of magnitude or 2 or 3 orders of magnitude less than what we've got here. So we just want to make our copy of DNA. We want to check it to make sure that we don't make any errors. If you go back to was it the medieval monks and they draw like the snails battling in the margins and all the stuff in there as well too. But every now and then they make a mistake and you could actually see where they could scrape the parchment to get the ink off of there. Thank you very much. Or do something else to fix that mistake. So our DNA, during replication, it has proofreading mechanisms, and it has actually several different mechanisms that can help proofread in there that will check for errors and repair them. And then the other thing that happens is we can take large segments of DNA and we can just move them. This is where we kind of mentioned the transposons at the end of chapter 24. So what we can do here is we rearrange the DNA. So we make an exact copy, but we just, change it around a little bit, change it up. It's kind of like, same furniture in your room, but I'm going to put the chair over here and the desk over here and so on and so forth. So that's sort of what we're doing there. And that can allow us to get some unique expression patterns. Okay. And as with all metabolism, there is a series of tightly regulated processes that govern this whole replication. Okay. So I think I mentioned last time that we're going to kind of divide things up between bacterial examples and eukaryotic examples. Bacterial tend to be simpler and a little easier to understand, and eukaryotic are similar, just with added layers of complexity in most cases. Okay, so to do that, we're going to do a little background information here as well too. When we talk about bacterial genes, we usually give their names as 3 italicized lowercase letters. So in this case, we'd have this little UVR that's shown up here, and this would be coding for the UV resistance gene. Sometimes we can add another letter on there to indicate that there are multiple genes involved. So we just kind of list them in order of discovery, like DNA A, DNA B, DNA C, so on and so forth. And those are replication genes. Now, the bad news is they typically the letters get added in the order of discovery, not necessarily the order of function. So there might be, you don't necessarily go in alphabetical order for the whole replication process. They might be in a different order there. They can spell cool stuff. Bacterial proteins, on the other hand, are going to have names that are often similar to their genes, but they are not italicized, and they capitalize the first letter. So we might have a lowercase italicized SHH gene that encodes the protein capital SHH, Sonic Hedgehog, for the protein in there. And you can see the example here. They have DNA A. It's encoded by the gene DNA A. I wish I could speak in italics. It's like I once typed a paper where every word was in a different font just to annoy my friends. Okay. So if we look at a typical E. coli chromosome, we can see that we have all sorts of stuff in here. You guys got to memorize this? No, I'm kidding. No. But we will look at some certain examples in here. We'll talk about a few of these things, often like our rec genes, where you think, hey, recreation, recombination and repair genes in there, replication stuff here, here's our origin, so on and so forth, all sorts of stuff in there. Just a nice big map of the E. coli chromosome, which being so small is, understandable. And so when you get into like the human chromosomes, that's where my brain just kind of goes, oh, I'm a protein guy. Okay. For eukaryotes, we don't have a universal system of naming them. So it can vary between species. I always laugh about the Drosophila people. I worked in a fly lab one time as an undergrad for a semester, and they're just like a different type of people. But that's where like the gene Sonic Hedgehog comes from and stuff like that too. But the fly guys have their own nomenclature, and it's just kind of all over the place. It's like whatever. So people have sort of tried to make universal rules, but there may be universal species rules in there.
So they have like separate rules
for just the way they name them. Yeah. So instead of having like that little 3-letter abbreviation, all that, sometimes you'll get crazier names and abbreviations in there. Yeah. Fly. And well, I guess I was gonna say, I don't know. I grew up. Oh. Yeah. Well, I grew up in a house that had very old brick walls, so we'd have flies that would find their way inside. So the wintertime when the sun would come out, our windows would be full of flies. Mm-hmm. So I would have had no problems murdering flies like crazy. But yeah, same thing with we had bees in our windows too. But oh well.
I love bees.
Yeah. These were yellow jackets, so not friendly bees. Yeah, mean bees. Okay. Thank you very much. All right. Okay. So we're just into chapter 25, and we're just looking at naming conventions. So eukaryotes, like I say, are a little bit different in here. Usually the name is going to refer to the function, unless you're like one of those Drosophila people where it's going to refer to whatever video game or anime that they were watching the night before. So make fun of the biology people. Yeah. Sorry. But anyway, for yeast, we typically the gene letters. It's going to be very similar. We're going to have 3 uppercase italicized letters followed by a number. So this will be our gene. So you can see the italics generally indicate a gene, non-italicized generally indicate a protein. So in this case, our COX-1 gene encodes for cytochrome oxidase, okay? Or at least a particular subunit of cytochrome oxidase. The protein naming really gets kind of crazy as well too, because we have so many proteins with and variations and isoforms and stuff like that. So we'll often see, we just take the gene name, right, and then add a P at the end of it for protein. So our RAD51 gene, italicized, becomes RAD51P for protein. Okay, but that's just our conventions of naming. So if you start seeing stuff, you guys probably read a few papers where you're like, what are they talking about? What are they talking about here? If you read lots of Cell, you'll see this over and over and over and over and over and over again. All right, but let's talk about replication, because that's the main focus of this chapter for DNA. All right, there are 3 fundamental rules. We have a semi-conservative replication. So it's like Florida? No, okay, yeah, sorry, political joke there. Yeah, but basically, when we make our DNA, we have 1 strand from the parent and 1 new strand, that's the daughter strand. So that's why it's semi-conservative. We keep one of the old strands, we have a completely new complementary strand in there. Okay? Replication starts at an origin and proceeds bidirectionally. So if we look at our E. coli circular plasmid, here's our origin. We're just going to start copying it both ways. Okay? Like my circle. Okay. And then the other thing is the synthesis of the DNA always occurs from 5' to 3'. In fact, you may have been in a lab where you've ordered stuff from a company called 5' to 3'. All right. So semi-conservative, this goes back to the measles and Stahl experiment, which was very simple and elegant. That terrible term that I saw used 3 times in a paper I read this past weekend. I was like, really? Too much. We're not in ball gowns here and tuxedos. But it was a very simple experiment, but it won a Nobel Prize because it basically showed that the old parent DNA gets split up and divided evenly between daughter strands in here. So we should have a nice little picture coming up soon. Okay. All right. So what they did is they proved this by growing the cells on a medium that had only nitrogen 15 in there, heavy nitrogen. So you get one band when the DNA was centrifuged in that way, because all the DNA that incorporated all that nitrogen in there, it was all nitrogen 15. So when you spin down the DNA, you get one band. Okay. So then you grow it on a new medium that has normal nitrogen 14 in there, and let it divide once, and you spin it down, and you see what you get. Okay. So, so here's our original one, which we spin this down, and here's our heavy DNA. So we come out in the centrifuge, ultracentrifuge, this one particular band. If you looked at things that were like this, here would be our light band, where it's all nitrogen 14. But here, when it replicates once, instead of getting 2 bands, a heavy band and a light band, what they got was just one band in here, somewhere in the middle. So this was our semi-conservative, our proof of semi-conservative, that we incorporated one daughter strand with one parent strand to get that in between there. Okay. So that's part of the way that we ensure that we have a high fidelity copy of DNA when we replicate it. So we keep a parent strand in there, and our daughter strand is just complementary to it. And then eventually that becomes a new parent strand for the next generation, and so on and so forth. And if you did something like this, like say PCR, you'd eventually see that you have way more daughter strands in here. So you'd have a whole lot of this light stuff, but you'd still have a little bit of hybrid DNA in there as well too. And you see that your heavy DNA band disappears after the first division because it just doesn't recombine. Okay. The alternate hypothesis was you basically unwind the DNA, make copies of each of them, then the old one would go back together and the 2 new ones would go back together. The problem with that, and that's where we'd see no intermediate, that purple disc on there wouldn't be there. The other problem with that is if you made an error in the daughter strands, you wouldn't know. They'd still match up as long as you made the error in both places in there. Okay. Okay. So here's what we're looking at. We take our DNA that we've packed in so tightly when we're ready for replication. We expose it, we open it up there, and we start making our 5' to 3' this way, and our 5' to 3' this way for our complementary strand. Okay. All right. So circular DNA goes both directions at the same time. It's bidirectional in there. Yeah, we're not going to worry about this proving in here, but basically just goes both directions. But that really means that you can get an origin of replication that will show up as a bubble. So if you have your circular DNA in here, and then you start at your origin of replication, let's see if I can do this. You unwind the DNA at the origin, right? So you get this little bubble here, and it'll expand all the way around until you've got 2 strands going, and they split off that way. Okay? Or better than like trying to imagine my fingers, something like this. Here's our origin of replication. We start off this way. We get our double strand here, and it splits off, and we have our daughter duplexes. Okay. I was going to say, I guess technically, if we wanted to be super accurate, we put one red one on the outside and one red one on the inside, but it's just a cartoon. And here you can see the replication forks, and here we can see this little bubble basically on the DNA. All right. So synthesis always goes 5' to 3'. So for one strand, that's awesome. You can just keep going the whole way there, 5' to 3'. But you can't go backwards. You can't go 3' to 5'. So the complementary strand is backwards. So what happens is you make a loop of it, and then you go 5' to 3' in the backwards direction until you get to the end of that loop. You release it, let it loop around again, 5' to 3', and just ligate those little sections together. And that's where we have these little tiny pieces that help ligate it together that are called Okazaki fragments. Okay, so we have a continuous replication on the leading strand, and we have a discontinuous one on the lagging strand, which has to go in the opposite direction here and have these Okazaki fragments that ligate it back together. Okay. All right. DNA is degraded. It's like, you suck. No, okay. Terrible. There are various nucleases that will degrade DNAs, and we call them DNases. Yeah, okay. They will target DNA and cleave it and cut it into small pieces. RNases, on the other hand, only degrade RNA. So that little extra hydroxyl group in there is very important. Exonucleases will cleave bonds that remove nucleotides from the ends of DNA, and endonucleases cleave them within a DNA sequence. Okay, so you often see restriction enzyme maps. Well, actually, I don't know if you'll see them too much anymore, but we can use restriction enzymes harvested from various organisms to recognize DNA sequences and cut at specific places. So things like BAM, H1, and PST1, and all those little New England biolabs restriction enzymes that you can buy to cut DNA at specific sequences. So they recognize that they're almost all endonucleases. On the other hand, rather than degrading it and chopping it up, when we want to build it by utilizing DNA polymerases. And so our polymerases, you can think of DNA as a big polymer, just like rayon or Teflon or something like that. It's a DNA polymer. We have multiple polymerases that are involved in the synthesis of DNA. Okay, so DNA polymerase 1 was first one discovered in E. coli. E. coli has also DNA polymerase 2, 3, 4, and 5, and they all have their particular roles. Okay, all right. As far as to how we actually make these, replicate them, our elongation chemistry is not too bad. We use our parental DNA as a template. We have a big pool of nucleotide triphosphates. So you'll have your ATP, GTP, CTP, and TTP in there. They'll serve as our building blocks. We take our nucleophilic hydroxyl 3' end. Okay, so that's a good active group in there. And it will attack the phosphate of the incoming trinucleotide. And we have to have this 3' hydroxyl group in there, or else it just won't work. And we make it more powerful by using magnesium as well too. I used to ask the question in Biochem lab about what's required for our polymerase reaction to occur. And so you got to have your polymerase, you got to have your template strands, you got to have your nucleotides. But one of the other things is magnesium. You got to include magnesium in there. But now we use a mix, and it's just, just add this little bead and your sample, in water. But anyway, but the other thing is pyrophosphate. That 2-phosphate group is a really good leaving group as well. So if we look at it, their strands, we can see, hey, this growing strand, this hydroxyl group can attack the alpha phosphate, and we can add in our T. So our adenine base pairs with our thymine here, recognizes these two. This attacks the phosphate, kicks off the pyrophosphate here. These magnesiums help stabilize this, hold it in the right position, and then we add our A to this, and we'll move on. We'll find a G that will come in here and match up with this, and the same thing will happen. We'll just slide on down the polymerase strand there. Okay, DNA polymerase requires a primer, so we got to have a short start to this. Okay, this is going to have a nice 3' hydroxyl group to start the reaction. It can be made of DNA or RNA. Most often it's made of an RNA primer that's in there. Okay, so our primer just kind of recognizes the DNA sequence and sits there and then says, hey, polymerase, come bind here. It's like the friend who finds you a parking space. Save this parking space. Stand in a parking space so you can drive into it. That's essentially what our primer is doing, which allows the binding and that initial DNTP to start there in there, and then our polymerase can start translating along there. Okay. All right. So the DNA polymerase has a pocket that has two particular regions in there. There's the insertion site where our nucleotides come in, and the post-insertion sites where we can move along the DNA strand. Okay, so if we look at this pictorially, okay, we can kind of think of this as a hand that wraps around the DNA. And so you've got an insertion site near the palm, and your fingers in your thumb. Got to do this. Let's see if I can do this the right way here. Yeah, I don't know. Flip something. Like this. There we go, wrapping around their way. Okay. And so you're basically kind of like if you think of like you're, letting a rope slide through your hands, or, a stick that doesn't have splinters in it. You can slide it along there, and then your palm is allowing the nucleotides to kind of hop in there. Okay. All right. So once DNA polymerase is bound and maybe adds that first nucleotide, it has two choices. It can slide along the strand and match up the next section and add another nucleotide. That's called processivity. It'll process along there. Or it can say, nah, that's enough, I'm done, and dissociate. Okay? So our different polymerases have different binding affinities for our DNA. And you'll see that, I think it's DNA polymerase 3 has the strongest affinity, so it'll stay on there and it'll process along for several hundred, maybe a couple thousand base pairs. I think it's DNA 3. We'll see here in a couple minutes in here. Others will have less affinity for the strand, and they may only go a few base pairs, and they may be involved in proofreading or repair, or in that reverse, that discontinuous part in there. Okay, so a few nucleotides to several thousand nucleotides at a time. This was actually one of the challenges in like the Human Genome Project. I remember being in grad school when this was undergoing its way. And you start off learning about polymerases, and it's like, well, we'll just take a polymerase here at the start of this chromosome and do the whole thing. Except that's, thousands upon thousands, hundreds of thousands of base pairs, and there's just no way it's going to stay on there. So the Ventnor and his lab used the shotgun approach, and they just kind of blasted the chromosomes into small pieces, replicated them, and then put them back together. So that's an easier way, a faster way to do it, but the challenge was putting all the pieces back together once you break them into tiny pieces in there. But our different polymerases, like one of the reasons why I did a lot of mutation work was I did that in E. coli or in plasmids. So in plasmids, we know the size of the DNA, double-stranded DNA in there, the circular plasmid. So it's short enough that we can use the right polymerase to just do the whole thing at one time. And actually, the plasmid I used was way too big to be a plasmid. I mean, it was like 2.5 times the maximum size of a plasmid from what we say today, but it worked. It just meant my PCR would take like a day and a half to run, because the longer the replication cycle, the more time you needed for each cycle in there. So it would take like a good 4 or 5 minutes for the elongation phase, and so it would take a day and a half to do that, but it worked. Probably shouldn't have worked as well as it did. All right. So again, if we look at our replication, it only makes an error in every like 1 billion to 10 billion base pairs. That's astounding. That's like basically hand copying the textbook, 100 or 1,000 times or something like that, and not making any mistakes at all in any of the letters, getting everything in the right order in there. And the reason this works in part is because those base pairs, whether it's AT or GC, those base pairings help it line up nice and easily. I guess two hydrogen bonds versus 3 hydrogen bonds. But there's also a lot of proofreading that goes on as well too. The polymerase active site will exclude base pairs that don't have the right geometry. Okay, so the neat thing though is polymerases, they're just going to do it. It's kind of like, you see the guys who are fixing the road, and it bugs me where they've repaved the road near my house. There's like a little blob of asphalt that fell off, and it's like, it's only like a little asphalt pancake in there, but it would have taken like 30 seconds to just hack that off of there and have a nice smooth road. But no, there's this little bump there, and it bugs me. It's like, it's good enough for government work, right? So that's sort of our polymerases. They make errors every 10 to 100,000 base pairs, but there's lots of proofreading and repair mechanisms to fix them as they go along. So there's actually like a little guy there who's not leaning on a shovel, but he will actually fix that little bump in your cells. All right. Again, you see our geometry lines up in here, A with T and C with G. Okay. So the errors can get corrected by a 3' to 5' exonuclease activity. So this is something that all polymerases will have this extra activity in here. It proofreads it and says, hey, did we get that one right? Yeah, you kind of check it and make sure it's good. Okay. Kind of like if you have to sign a whole bunch of documents and then you stack them there, signature on there, okay, signature on there. So you're just kind of checking to make sure everything's in order. Okay. It won't move forward until we go check. So it's kind of like if you're trying to like say change your schedule, you got to submit stuff, right, and somebody's got to sign off on it. Yeah, you ever have that frustration? It's like, who in this office is not checking off on this? Yeah, right. Where's my advisor? So I try and be fast. But all right. If there is an error, it will pause it, and we'll actually see that it will cut out that little section in there, and it will remove that nuclease and allow the correct one to be inserted. Okay, from that 3' to 5' exonuclease activity. It's like, oops, we put the wrong one in there. Let's kick it out and do that. Okay. So I mentioned there are at least 5 DNA polymerases in E. coli. DNA polymerase 1 is abundant. There's a whole bunch of it, but it's not the major one. It's just the first one that was found. It will read about 600 nucleotides per minute. So that is too slow for that replication fork because we actually time that replication fork and see it go faster than that. It has very low processivity. It doesn't go along there very far. So it's often used primarily for cleanup. DNA polymerase 3 is the primary replication polymerase for E. coli. 2, 4, and 5 are basically repair polymerases. So if you make an error, they go in and they're the ones that help fix it. All right. Okay. So here's what they do. You can look at the various sizes. Yeah, I love this. These do have different names. I know, because they were discovered by different groups working on different stuff in here. But that's our major one here. Our exonuclease activity in here. You notice that 1, 2, and 3 have this 3' to 5' exonuclease activity. 4 and 5, not so much. And only 1 has 5' to 3' exonuclease activity. So these are very specific, but they lack that activity. They're not primarily involved in replication. This may be a different mechanism of repair that's in here. You can also see how fast do they go. 10 or 20 nucleotides per second, 40 nucleotides per second. I love this one, 1 nucleotide per second. This is our artisan, handcrafted, bespoke polymerase in here. We're going to make sure we get this exactly right, but we're going to go really slow, as opposed to polymerase 3, which, if you think about how fast bacteria grow, 250 to 1,000 base pairs per second. Just flying through there. Okay. All right. DNA polymerase 1 also has this 5' to 3' exonuclease activity. It moves ahead of the enzyme and helps hydrolyze things that are in the path. It can also do a little nick translation. So it can break the DNA strand, and then that nick will move along with it here. So this is known, these are basically in this clinal fragment. So we can have this little particular domain that gets separated ahead of it. Okay. But the important thing is here, our polymerase 1 is actually cleaving this little section here. We slide it along. So here we have our monomeric nucleotides. Here are the ones that we're adding in. Okay, so we're creating our new strand and we're removing these old ones. Okay, so if we're cleaving off those old ones, clearly this can help be a repair mechanism. Okay. Polymerase 3, really complex. It's got 10 subunits. And keep in mind, this is an E. coli. When we start talking about eukaryotes, ooh, crazy. All right. I love this. The core domains are linked by the clamp loader complex tau2 gamma delta delta prime. That's just indicating the units that are involved in there. And we're not going to sweat that too much. The important thing is there's actually this nice little clamp that helps prevent dissociation, which is why the processivity for polymerase 3 is so high, because it basically clamps on there, slides along for a long distance. Hopefully, presumably, the entire E. coli chromosome in there. And if we look at this in kind of a cartoon form in our space filling model, you can basically see that, hey, we have this nice little swinging clamp in there. It's going to come around there and latch on to our DNA and move on along. Okay. Is all this okay so far?
All of this was in my genetics course except for
Awesome.
one now.
Yep. And I was going to say, those of you who have had some of the biology courses, this should be like a refresher. Yeah, so that's good. That's why I don't feel bad about it. This is where you get to breathe from going, oh my God, genetics to, or sorry, oh my God, metabolism to, genetics. Okay. Yeah. Lots of different subunits in here, the things that they're involved in, the particular gene that makes them, all that stuff in there. We're not going to break it down that far, but the key is we got a whole bunch of subunits, and this is a really complex. Keep in mind, this is a bacterial enzyme, complex. So we're making it a very complex thing for bacteria. All right. So what do we need for bacteria replication? All right. Well, so E. coli takes over 20 different enzymes and proteins that are involved in here. We can call this the replosome. So we have our proteome, we have our genome, here we have our things that are involved in replication. We're gonna call that the replosome. So we gotta have our helicases, unwinding our DNA. So we gotta, basically expose that DNA so it's not packed in there tight together. We gotta open it up so we can actually, replicate it, see it there. So our helicases in there, our topoisomerases that are going to affect that linking number, relieve the stress of the unwinding in there. We're going to unwind from unwinding. Okay. Our basic DNA binding proteins that will stabilize those strands so they stay apart because they're going to want to come back together. Our primases that'll make the RNA primers to get them started, and our ligases that will seal any nicks in there, particularly for that discontinuous lagging strand replication. Okay. So we got three things that go on when we do replication: initiation, elongation, and termination. So how do we start? How do we keep going? How do we finish? All right. So our initiation begins at the origin. So the ORC site in here. Typically for E. coli, this is a particular sequence that is 245 base pairs in length. Not that you need to know that, but you just need to know that it starts at the origin. And it will have a highly conserved sequence in there. So we're going to see these particular base pair repeats in there that serve as a signal for the binding of this initiator protein. Okay. And once that initiator protein recognizes a site, it can bind there. It will include a couple other components as well too. So here we see our origin sequence, some of the domains that they contain in there. And we'll have, like I say, at least 10 different proteins that are involved in here. The whole goal is to open up that helix, get it ready, and form this pre-priming complex. All right, all sorts of stuff that goes on in there. So we're going to have these, ATPases, DNA wraps around the complex. We have a nice positive supercoil that's going to induce some strain. So it's going to denature nearby sites, those do sites from that origin sequence up there. All right, when those denature, then other proteins will recognize those sites and bind, and they'll help facilitate that DNA bending and enhancing that strain and exposing making the double strand into single stranded. So we'll see this hexamer structure gets opened by the binding of DNA C. That's probably our first big key there too. And then DNA B will migrate along the single stranded DNA and start unwinding it. So that's working as our helicase in there. Okay. Then other proteins can bind to DNA B, particularly our DNA polymerase 3. Okay. DNA gyrase it's going to be one of our topoisomerases that will relieve the stress ahead of the replication fork. So here's what we see going on here. So here's our sequence in here. We get this kind of exposed. We have these DNA A's bind here, or DNA B will bind in here. We can kind of stabilize this this unwound portion here, and then we get our helicase in here as well too. and then DNA-C can bind, and then that's our trigger for DNA polymerase to bind. This is way more detail than we need at this point. So initiation will occur once per cell cycle, right? So basically, we're going to start this up, make a copy, and we proceed into a new cell cycle. Time scale is 20 to 40 minutes for E. coli. Okay. So how do we regulate initiation? Okay, this is going to be involved with methylation patterns. So we have damn methylase, or as I referred to it as I started studying all this stuff, it's like that damn methylase. Which is just short for DNA adenine methylase. It's going to add a methyl group to the nitrogen of adenine and the GATC sequences in there. So these can interact with the plasma membrane. Keep in mind, our nucleosome is membrane adjacent, if not bound, in bacteria. So we see this kind of localized nucleosome structure in here that's kind of quasi-attached to the membrane in here. All right. And then once we actually make that replication, we'll see that this will dissociate from the membrane, and then our methylase can fully methylate the daughter strands as well too. So one of the purposes of the methylation is to tell the parent from the daughter. All right, so I don't know if you ever seen like those Instagram photos, like which one's the mother, which one's the daughter? I don't know, cheesy stuff like that, right? So for DNA, if you make a mistake, in the daughter, if the daughter, I've got a mistake in there, we got to fix that, right? We'll send them to boot camp. No, sorry, they can't do that in an E. coli cell. But what they can do is they can, cut out the wrong sequence and put in the correct sequence. How do they know which one's the wrong sequence? Well, the parent strand will be methylated. Okay, so the proofreading mechanisms will recognize the pattern of methylation and say, okay, this strand is methylated. This must be the correct one. We'll cut out the sequence on the daughter strand that is unmethylated. Once you've completed replication, then the daughter strand will become methylated as well too. So that during the next cell cycle, we can start the whole thing over again. Okay. So we've got our initiation. We can start now into elongation. For the leading strand, it's nice and simple. We take our primase, which is going to make our RNA primer. Our DNA polymerase 3 will attach to that, and we'll start adding nucleotides to the 3' end of the strand. Eventually, that RNA primer will be degraded as well too, but we'll make our copy there. So our DNA polymerase just processes along, keeps on adding nucleotides, 1,000 to 2,000 nucleotides per second. Okay. For the lagging strand, it's a little more complex because we can't go through 3' to 5'. We have to go 5' to 3'. So we're going to have to kind of make a loop of this to run that through the DNA polymerase to add its nucleotides backwards. We're not really going back. We're going forwards, but we're twisting around the DNA, the lagging strand, so that it's running through backwards so that we can add 5' to 3'. And then we have to detach, straighten it out, make a new loop and add it in there and then ligate those two together. Okay, so we make this nice little loop. Okay, so this is not necessarily the easiest thing to sit here and look at and understand. So here's our leading strand, nice and simple. We're just chugging along, everything's all well and good. For our lagging strand, we have all these other little helper proteins that will bind there. Got our RNA primer in here, kind of go back this way. So what we're going to do is, here's our lagging strand. We run this through this way and then run it in backwards, loop it around and run it in backwards, so that we're constantly making this go backwards. Okay, and then we have we get our Okazaki fragments in there, and then we ligate the ends together. Okay, so we just kind of twist it out that way. All right. So leading and lagging strand synthesis, you can see here we have our cartoon of everything together in there. Our clamp that opens everything up. Taking our lagging strand here and then running it around this way. This is going this way, so now just pull that through backwards. I don't know if you can see that as well. It took me a long time to look at that and see, okay, that is backwards. Is that backwards? Go forward in there. So we're not going to sweat it. Okazaki fragment nears completion. We get a new primer that binds on there, and we ligate that together, and new beta clamp in there, and so on and so forth as we go forward. Okay, and then we can just basically, So as for each segment, each Okazaki fragment that's in there, we leave our beta clamp behind, put a new one on, do the same thing to run it through backwards again. Okay. So that's kind of how that works. Okay. Yeah. Awesome. Okay, transitioning between those. So the RNA primer gets removed by DNA polymerase 1, or sometimes this RNase as well too, usually RNase H1, and then polymerase 1, pol 1, will fill in that gap, and ligase will seal the nick, ligate it back together. Okay. All right. Final steps in the synthesis. We can basically get rid of our primer, ligate everything together, and now we've got a parent strand and a daughter strand. Yay, we're almost done. Right? We got to terminate. How do we know when to stop? Okay. DNA ligase will make that bond between that 3' hydroxyl and the 5' phosphate group in there. Again, those are good active groups in there, so they're ready to. The enzyme just facilitates that reaction so that we can link these two together to make our release our pyrophosphate, and we seal up our DNA. Okay, and we use ATP for this as well too. All right. Our last step, termination. How do we know when we're done? The bell rings. No, okay. So our replication forks will meet at a particular region that has 20 base pair sequences, our TER sites, which will bind TUS. I don't know. Yeah, that's the naming. All right. So we have all this stuff in here, and we basically end up making these little fork traps. So when these forks interact with each other, that's when it says it's time to stop. More complex than we need to know. Like I say, if you go to graduate school and start looking at this particular type of stuff, you will get into this level of detail. But basically, they interact and they say, "Okay, you're done. You finished it. You're done." Okay, take a deep breath. Right. So that's bacterial or prokaryotic replication. What about eukaryotic replication? It's very similar. We're just gonna add a whole bunch more proteins in there and more layers of regulation. Okay, so it's a lot more complex. You got to think our eukaryotes have a lot more DNA, so they can't replicate quite as quickly unless we have multiple sites of origin, like yeast that will have 400 well-defined origin sites as opposed to one for our E. coli. And these are called our R sites, autonomously replicating sequences. The entire genome gets replicated once per cycle. So of course, we have to make sure that all these sites are coordinated and we're making one copy each time and then putting them together in the right order or transposing them properly in here. So these are going to be our cyclin-dependent kinases and our cyclin proteins that help regulate this so that the entire genome gets replicated once per cell cycle. And then the cyclins get ubiquinated at the end, so they don't say, "Oh, do it again, sneak in another one." No, it's kind of like you've got a ticket for something. Use it once and then it's void, right? You can try to use it again. Nope, it's been ubiquinated, so you can't do it twice. No scamming your way back in for a second helping or something. All right, so let's compare our initiation of replication. We have this pre-replicative complex, or pre-RC in here, and that ends up making an origin recognition complex, or an ORC. I don't know if you guys like Lord of the Rings or Warhammer or Dungeons and Dragons, anything like that, but the orcs are coming, time to replicate the DNA. I don't know, it doesn't seem to play into fantasy very well. But basically our ORC functions much like the bacterial DNA. Okay, it's just more complex and at more sites. Okay, so we can see that our ORCs will bind to the origin DNA, and then they'll initiate the binding of all these other cyclin-dependent compounds and so on and so forth. And we see our whole thing going here. The eukaryotic rate of replication is slower than bacteria. I know you've probably seen that 5-second rule. If you drop the food on the ground, how fast can you eat it? Or if you're at a picnic and a potato salad's been sitting out on the table in the sunlight, how long can it sit there before you can eat it? Because bacteria grow so fast. Eukaryotes much slower. So as opposed to 1,000 nucleotides per second, we might see about 50 nucleotides per second. Much slower procession in there. But the thing is, we have origins every few hundred. every 30 to 300,000 base pairs. So we have a lot more origin sites than E. coli. So we got a lot more of this polymerase activity going on simultaneously. Okay. All right. And again, just like E. coli has 5 different polymerases, there are lots of different DNA polymerases for eukaryotes as well too. And instead of calling them polymerase 1, 2, 3, 4, and 5, we call them alpha, beta, delta, gamma, epsilon, so on and so forth in here. Okay, so Pol alpha is our primase activity in one subunit and polymerization in another. It does not have the 5' to 3' proofreading. Polymerase delta is for the lagging strand and epsilon for the leading strand. Let's see here. Both are very processive. These, polymerase delta and polymerase epsilon are comparable to polymerase 3 in bacteria. So those are the heavy lifters for the replication. And both of them have that 3' to 5' proofreading. So they won't process if they make an error, in theory. As far as termination, here's where our telomeres come in. You get to the end of the chromosome, you recognize the telomeres, and says basically, okay, you're done. Okay, we'll talk more about telomerase, which uses a RNA model in there. So we'll talk about that in the next chapter, sometime next week. Okay, all right. DNA polymerases. So if we look at the herpes simplex virus, it encodes its own polymerases. So they have their own unique genetic code, and we can target their activity by the use of acyclovir. Okay, so our cyclovir compounds can target that and inhibit the herpes virus from proliferation. Let's look at repair and mutations. Okay, so if you look at something like Parkinson's disease arises from a particular mutation. Actually, it's in mitochondrial DNA, so we have a slight, it may not be quite as relevant to this as we want, but how does a mutation get through if we have all these repair mechanisms? All right, so we can look at oxidative damage. It basically comes down to an accumulation of damage. If you make one error, that's not going to be too bad. 1 or 2 or 3 errors, not too bad. But if you have 1,000 errors, then how do you repair that much damage? Okay, it's kind of like you break a window, you can replace a window. You knock out a wall, you're like, oh crap, how are we going to fix that? Okay, blow the roof off or something. So the majority of damage, mutations, or errors or whatever are corrected using the undamaged strain as a template. Some base changes will escape repair. Sometimes an incorrect base can still function sufficiently well, and it will work in replication as well too. So if that happens, then the daughter strand is going to have a change sequence in both strands ultimately, and you'll see that move along. So an accumulation of mutations in eukaryotic cells gets correlated with cancer. Most carcinogens are also mutagens. So if you read like the warning labels on some bunch of chemicals in lab, not that we're ever in lab, you can see that they'll list as carcinogens, teratogens, mutagens, all that good stuff in there too. So typically we get thousands of lesions per day, and these are unrepaired DNA damage, but only 1,000th of them, 0.1%, will actually become a mutation thanks to DNA repair efforts. Okay. And we have 130, at least 130 repair proteins that are involved in fixing the errors that get made. So a lesion is DNA damage. If it doesn't get repaired, it becomes a mutation. Okay? Our mutations can be substitutions, particularly point mutations where we just change out one base for another base. They can be deletions where we just cut out some bases, or it can be an addition where we stick in some extra ones. Those latter two you'll typically see if you have like a run of a bunch of the same base, like A. It's like, was that 7 or 8 A's? Like Dirty Harry, was that 7 bullets, 8 bullets? So I forget. Whatever the movie was. How many bullets do you feel, lucky punk? Yeah. All right, but
my niece has a 1p36 deletion in her DNA, which causes her to be not be able to talk.
Okay.
And also some other delays and everything.
Yeah.
Why doesn't it catch itself and fix? Because that's not an inherited disorder, that's an actual mutation while.
Yeah. Yep.
So why doesn't it fix itself? Like, I don't get that. Like, because a child will not have enough oxidative stress to cause that mutation.
Yep. So the short answer, the really, really short answer is math. It's just a statistical luck of the draw. Most of them are going to get repaired, and most of them are going to be caught, but every now and then one will slip through. With the fact that 98.56% of our DNA doesn't code for anything, the ones that slip through often don't mean anything. So it's just a really extremely rare occurrence that something important gets mutated and slips through. I'm gonna have to look up more about that IP3 because say the nonverbal portion. I've got a friend whose daughter has it's some mutation, but it's really rare because only, well, at least in the West, only 30, maybe 36 people have been identified to have this particular type of deletion. And they're all nonverbal and have particular sets of delays, and it varies between them as well too. Some would depend on whether they're homozygous or heterozygous. Affects the severity of it, but it is a frustrating disease because imagine.
But deletion doesn't come from the parents, right? It's not.
Correct. Well, if the parent has a deletion, and they can pass it on because that changes the template in there too. But the deletion can happen just whenever you have like a series of bases that are all identical, and you're like, did I copy 7 or 8? Oh, 7 sounds good. Same way to add another one in. That'll often happen.
If a parent had a deletion, is it possible that deletion didn't show up like phenotypically, and then it showed up? It is possible.
Yeah, I think so. Yeah. So you get silent mutations that are someplace that are, maybe, and even if it's in something important, if you just have a heterozygous copy, a heterozygous deletion, maybe the good copy is still sufficient to. So it's kind of like sickle cell in that regards. If you're heterozygous, you're generally won't notice things unless you're at like really high altitude or under a lot of stress or something like that. But if you're homozygous, then the effects are often much more severe. So our silent mutations, again, will have no effect on the gene function, or it doesn't affect a region of the DNA that's involved with genes. Okay, so these mutations, like I say, it's like the draw, and we get several thousand mutations a day. That most of them are meaningless. It's just in non-coding stuff, and many of them are repaired. It's just and then some of them. The reason that some of them happen is, again, evolutionary pressure. It's like, well, let's try changing this slightly. Is it better? You think of the optometrist, better like this or better like that? It's like, there's really no change, no significant change here. If we look at the Ames test, this is just basically a test to figure out how mutagenic a particular compound is. So in this case, they used a particular Salmonella strain that makes it unable to synthesize histidine. So they add a compound to it to see if it'll grow in this histidine-free medium. And basically, here's what we see, something that we got here. So this is our regular Salmonella plated on a histidine-free medium. Shouldn't grow. Few of them do grow because they end up with a back mutation that puts the ability to grow on histidine-free medium in there. If we look at B, B, C, and D all have equal numbers of cells in there. We have our parent colony plated there. And we have a disc with various concentrations of a compound in there that will cause mutation. And you can see where these things grow at, okay? So as the mutagen diffuses away from this little piece of paper in the center, okay, it gets diluted. And you can see that in this case, in D, the mutagen is not at a sufficient concentration, or it doesn't have any effect on the growth of the cells. Here, it creates enough of a mutation that they can't grow at this distance, but you get beyond that, and they can adapt to it, and it's fine. And here you see a stronger mutagen where that distance, that no growth zone is much greater. Okay, so they're not inducing or they're basically have a much higher mutagenic potential. Okay. Yeah. Okay. So the more we get back mutation, the more that they will grow there. Okay. All right. If we look at types of DNA damage, you can have mismatches. So you can put the wrong nucleotide in there. You can have abnormal bases where instead of A, T, C, or G, you get something else in there. Maybe it's a methylated or adenylated compound in there. You can get pure pyrimidine dimers. Yeah, these are a little more serious in here. This is from UV light. And then you can get backbone lesions from ionizing radiation and various free radicals in there. And those are very challenging for the body to repair, which is like radiation exposure. You kind of, oh, it's fine, it's fine, it's fine. Boom, you hit the threshold, and now you got damage and radiation sickness and so on and so forth, and potentially death depending on the amount in there. So again, it's a threshold type exposure. Here are various enzymes that are and proteins that are involved in different types of repair mechanisms in here. Just know that there's lots of different ways to that. And again, we look at our mismatch repair relies on methylation. The parent strand will be methylated. The daughter strand has not encountered the damn methylase yet. So it will be unmethylated, and the repair mechanism will trust that the methylated parent strand is the correct version. Okay. Yeah. Okay, methylation has basically we're talking about the actual mechanism in here. More than you need to know, at least for now. Yeah, mismatch repair. Okay. If we have a mismatch repair, again, kind of the same deal here. We check and see for the parent strand in blue with that methylation pattern in here, and then we will cut out the mismatch here. See, we make that nick, and then we can cut out a section, and then we can put in back the correct section. Okay. There we go, something like that. Okay. And I guess there's two different ways that you can do this. Okay. Anyway, yeah. So we cut that out. Use exonucleases that chop off that little region and rebuild it with our polymerases, and then seal it with our ligases. Okay. Basic scission repair is going to look at these glycosylases. They're going to recognize particular lesions in the DNA, and they will cleave that nitrogenous glycosidic bond between the sugar and the base. So you get an apurinic or an apyrimidinic site. You basically just have the sugar there without the base. Okay, I guess we call it an abasic. I don't know. So and then we have particular glycosylases in there, like uracil glycosylase and all that. So sometimes you may see uracil getting incorporated into DNA. So it shouldn't be there. So we'll have this uracil glycosylase that'll remove the uracil. And the reason this happens is, it may be a cytidine at the time, but it spontaneously deaminates to give you the uracil in there. There we go. I like this too. Yeah, you'll note that the deamination is 100 times faster in single-stranded DNA. So again, another function of double-stranded DNA is a protective function in there. So that's so much stuff that you can do with that. So for repairing sites in bacteria, you remove the entire nucleotide, not just the damaged base. In fact, sometimes you'll just clean up everything inside. So again, going back to my road analogy, if you fix a pothole, sometimes you'll just dump in some stuff and try and level it out, right? But other times, if the pothole is too big or a crack or the underlying foundation of that, they'll just take out a big section of road and repave that whole section of road. And then kind of in between, and they're removing the whole thing is like the sides of the roads where they repave the sides of the roads where they've kind of crumbled a little bit. So those would be our analogies in there. And again, we'll use polymerase 1 to help fill in this little gap, and ligase to reseal it. Okay. So you see here we have our incorrect base. We remove our base. We'll cut out this little section here, put in our new stuff, and ligate it back together. Okay. If we have really big lesions that are occurring in there, then we'll look at a nucleotide excision mechanism. So we're just gonna, that's like our road paving. We're just gonna cut out a big chunk of road and repave this whole little stretch. So that's what we're doing. We're cutting out a whole bunch of stuff in here. We use this, it should be exonucleases. Exonucleases? I don't know about that. Yeah, these exonucleases will cleave the backbone into two different places, and they'll take out the incorrect sequence in there, and they'll use polymerase 1 and DNA ligase, just like all the others, to fill it back in. Okay, so here we have big lesion. Okay, so for E. coli, we'll just cut out this big section here and then fill it back in with polymerase 1 ligase. Very similar mechanism up here, except we cleave out even more, and we use our epsilon and our ligase to fill that back in. Same overall stuff. It's just in humans, we tend to cut out a lot more to make sure that we've got the bad stuff. I don't know. Okay. Gotta look here. Yeah, I think we can do a couple more slides. For direct repair, there are some neat mechanisms here as well too. So if you get that UV light that causes dimerization, we have photolyases that can repair those dimers. So these aren't found in humans or pretty much any mammals that have placentas, but it's kind of a neat way that fixes this dimer. For us, we have to actually like cleave it out with an exonuclease, but we're not going to worry about this. It's just another type of repair mechanism. Okay. All right. So I think this is probably a good stopping point. I wanted to, yeah, this is a good stopping point. We'll finish this up on Monday, okay? And then we'll jump into RNA metabolism, which is going to sound a whole lot similar. We're going to be talking about bacteria and eukaryotes, initiation, elongation, and termination. And then what about errors in RNA? But it won't be quite as awful as this, because the next 50 slides here are all about mutations and repair and stuff. Okay, error, yeah, I like that. And recombination. I can't forget about recombination here. Talk a little bit about that. Anyway, I think that's enough. It's stop sharing.
What time is final grade? What day is final grade?
I believe it's either the Monday after your exam. Wow. What?